Welcome, everyone, to session 1 on day 2, entitled International Update on Early Age Onset Colorectal Cancer Research. To this session, we welcome with gratitude Associate Professor Dan Buchanan, who's the head of the Colorectal Oncogenomics group at the University of Melbourne. Dan is passionate about research leading to the prevention of bowel cancer and will provide an overview of the emerging global research on early onset colorectal cancer. This session will not have a Q&A, but please, if any of you have any questions or comments that you would like to discuss, reach out to the team at Colorectal Cancer Canada. Dan, over to you.
Hi everyone, I'm Associate Professor Dan Buchanan from the University of Melbourne. Thank you very much for the opportunity to present some of our work today on understanding the causes of early onset bowel cancer. Just as a bit of an overview, I'm going to talk about some of the statistics around colorectal cancer in Australia, and particularly around the rising incidence of early onset colorectal cancer that we're seeing. And then I want to talk about some of our research into understanding the causes of early onset colorectal cancer, and in particular show you our work relating gut bacteria as a new cause of early onset colorectal cancer. And then I'm going to finish by talking about some of our national activities and even some of the national activities that are focused on understanding the causes of early onset colorectal cancer. So in Australia, colorectal cancer is the second deadliest cancer. It's the fourth most common, and in the last few years we've had about 15,000 Australians diagnosed with CRC each year, which has led to about 5,000 deaths from what is a preventable cancer. Anatomically, as you may know, the large bowel looks like this big question mark, where we normally break it up or describe it as being the right side of the colon, as you can see here, or the proximal colon, or the left side or distal colon, and the rectum. So we know that if you can detect colorectal cancer early, it's very survivable, and even better than that, we have colorectal cancer is one of the most preventable cancers, and early onset colorectal cancer or young onset colorectal cancer diagnosed under 50 or under 45, depending on the definition, is become a really important area of understanding it for public health. In Australia, the incidence of early onset colorectal cancer, we have around 2,000 diagnoses per year now, which accounts for more than 12% of all the colorectal cancer cases, and normally that's in a roughly 50-50 ratio with males to females. In this figure here, these graphs here, we've got 3 different age groups, so 18 to 39, 40 to 49, and over 50 group. And the top graph here is showing the number of cases diagnosed per 100,000 people in the population for 7 common cancers, where pink is bowel cancer, and in the bottom panel here, this is the cancer-related mortality by these same 3 age groups, again where pink is bowel cancer. So you can see in our 18 to 39 age group that, since the late 90s, early 2000s, we're getting this very sharp rise in the number of early onset bowel cancer cases. This is quite flat in the 40 to 49 group, and we know that in our over 50 group we're seeing this decrease in incidence, largely because of the National Bowel Cancer Screening Program, and this dotted red line here, vertical line, indicates when the National Bowel Cancer Program started in Australia. So you can see it's had an effect on reducing the incidence, but also reducing the mortality related to colorectal cancer in our older population. In the younger population now, this pink line has overtaken breast cancer as being the number 1 cause for cancer-related deaths in people under 40 and even now under 50. So these are very alarming statistics for us in Australia, and our colleagues, Professor Mark Jenkins, has calculated the 10-year projections. So we expect this trend to continue which will relate in another 1,500 deaths and $2 billion in lost income and over $1 billion cost to the healthcare system if we do nothing, which is exactly what is happening now at the moment. We have no strategy or no plan in place to prevent this this continued increase in deaths from early onset colorectal cancer. Sadly, in Australia, we also have one of the highest incidence rates of early onset colorectal cancer. You can see here at 13.5 compared to the USA and Canada. So again, this is really alarming for us and something that we need to really address. We have quite a limited understanding of what the causes of early onset colorectal cancer are. So if this ring here represents all colorectal cancer, we've got two rings. So we've got the outer ring, which is reflective of what we know now about the causes, where this purple part, we can definitely attribute 16 to 25% of the early onset colorectal cancer cases to an inherited cancer syndrome. So that's inheriting a DNA mutation from one or both of your parents. But you can see for the vast majority in the orange here, we don't know what the cause is. The inner circle here is related to the cancer tissue itself, and there's a routine test that is performed in most countries for every colorectal cancer that comes through the pathology service. So they look for a marker of what we call mismatch repair deficiency or microsatellite instability. And when that's there in early onset, we know that's around about 15% of cases. And detecting that's really important for two reasons. Firstly, it is a biomarker of potentially Lynch syndrome, and that is one of the most common inherited cancer syndromes here. But also in more recent years, it's become a very good biomarker for effectiveness of immunotherapy treatment. So the take-home message from this is that the majority of early onset colorectal cancer is mismatch repair proficient, so not amenable to immunotherapy, not related to Lynch syndrome or other inherited cancer syndromes. And this is the group here in the orange and the blue that are underlying the increasing incidence of early onset colorectal cancer we're seeing in Australia and globally. So in our lab, we're focused on really understanding what the causes of early onset colorectal cancer are. And I've just got 4 of these programs that we're investigating at the moment. The first one is around a genetic predisposition. So something that you've inherited in your DNA through either mutation or variation that increases your risk to getting early onset colorectal cancer. So I mentioned the inherited cancer syndromes before. And we know that current testing can potentially miss some of those. So we've been doing work in that space to improve the diagnostic accuracy. We know that there are likely to be some new genes that underlie early onset colorectal cancer. So we're doing some research to identify what those are, and also doing some work to look at polygenic risks. So multiple DNA variation that occurs within someone's entire DNA that puts them at an increased risk. So we're exploring that to help understand or detect people in the population who might be at risk. The second program of work looks at what we call the mutational processes. So these are the changes in DNA that contribute to development of early onset colorectal cancer. And when you look at all those mutational processes together, it's a function we call tumor mutational signature profiling, and it can help to tell you how that tumor is developed, but also can provide insights into what the causes have been, and I'll show you some examples of that. A third program of work looks at the microbiome, that's the within the gut. So that's looking at the particular types, communities, composition of the bacteria, viruses, etc., that make up the microbiome in your gut. And more specifically, we're looking at certain strains of bacteria or types of bacteria that have a genotoxin, that create a toxin that is damaging to DNA, which I'll talk more about. And our fourth area of research is to understand how early onset colorectal cancer develops. So normally, most colorectal cancer develops from a normal colon into a precursor lesion called a polyp, and then extra DNA changes and mutations happen that drive it then to become a full-blown cancer. So by understanding the precursor and investigating the precursor polyp lesions, it can help give us clues around what have been the causes of that particular cancer. And the first bit of work I'm going to talk about is the integration of these mutational processes with the microbiome. So we're fairly certain that modern day early onset colorectal cancer, so the group that is rising compared to the 1990s, is a result of modern day risk factors. So things that have become new or become more prevalent in society, in our environment, have likely contributed to an increasing number of cases. And you can see for each of these, there is some evidence around these contributing to increasing rates of early onset colorectal cancer. Well, what we think is happening and is that for each of these, they're being mediated by changes to the gut microbiome. So we think we know that obesity can change the composition of the gut microbiome, so can diet, so can a sedentary lifestyle and red meat, and also antibiotics, as we know. So we think that these modern day risk factors are contributing to the development of early onset colorectal cancer by mediating and changing the gut microbiome. So that's something that we're investigating at the moment. So throughout the large bowel, there are regions in there that we call biofilms. So these are mucousy, slimy layers of that cover the colon skin, essentially, or the epithelium. And they can be a home for lots of different types of bacteria. They can attach to the surface of the colon and help with the increasing the leakiness of the colon's barrier, so that enables bacteria and other things to infiltrate into the cells and then into the DNA. So we're looking at the moment to try and measure these biofilms and their relationship with early onset colorectal cancer here. So this is what it looks like under a colonoscopy view. This is under a microscope slide. So the blue area is part of an early onset colorectal cancer, and this red magenta color here is staining for a particular biofilm. So as I mentioned, we think that they're homes. They're really a hub for bacteria to cultivate and grow. But that may also be enabling genotoxic gut bacteria to grow. And there's 3 types of bacteria we're particularly interested in at the moment because they have a previous association with colorectal cancer. So one of them is Fusobacterium nucleatum, the other is Bacteroides fragilis, and the middle one here is a strain of E. coli called PKS positive E. coli. So each of these are interesting because they each produce a genotoxin that can be damaging to DNA or to cell function. So normally people have shown these associated with colorectal cancer by measuring them in the stool. But what we've done is measure the presence of these bacteria in the cancer tissue itself. And this has shown us that across 1,700 colorectal cancers, about 10% of the colorectal cancer tissue have the presence of PKS E. coli there. And you can see the proportions for these other bacteria. And what we're able to show is that they're not just present in any given colorectal cancer, they're present in certain bacteria in certain locations in the colon, on the right side or the left side, and also present in colorectal cancers that have certain types of DNA mutations. So this is giving us clues about how they might be contributing to the development of colorectal cancer. So this one particular strain of E. coli. So E. coli is very common in people, but there's one strain that has a group of genes called these polyketide synthetase group of genes, which indicated up here. They're important because they produce a genotoxin called colibactin. So what we think is happening is that the PKS E. coli are being allowed to be colonized in the colon through these biofilms. They infiltrate into the cell, produce their colibactin, which then damages the DNA. So it's not just the damaging the DNA that's important, it's damaging the DNA at certain places in our entire genome. So our entire sequence of DNA, it has specific locations where it causes damage. And this is represented in this little cartoon here. It binds in between these two strands of our DNA at specific regions. But what that means is it creates a very particular pattern of DNA damage, which we can measure then. So when we see damage in these certain areas of our genome, we can pull that together, and it creates what we call a fingerprint or a signature of the colibactin DNA damage. So this has really become really important in recent years. So how do we measure the DNA, the damage to the DNA? Well, typically for us, in our research, we take DNA from the tumor tissue itself and DNA from the person's blood. We perform a process called whole genome or whole exome sequencing, and we look for all the changes in the DNA that occur. So the changes in our blood that occur naturally, and then we measure those against the changes that occur in the new changes that have happened in the tumor tissue. And those new changes then we accumulate together and determine what sort of patterns they're occurring in. And this is an example here. So there's one particular type of pattern we call signature 1, another signature 2, signature 3. So when you measure these within any particular tumor, you build up a profile. So one tumor may have multiple different types of signatures in their profile, and that creates a very unique fingerprint for any particular type of cancer, early onset colorectal cancer. So this is a representation of those fingerprints across a whole range of early onset colorectal cancers. So each line represents a profile, a mutational signature profile for one cancer, so a unique fingerprint, and across here is all those colorectal cancers combined. And what you can see when you start to bunch them together by the types of patterns that you see based on, in this indication, based on the colors that are present, you can see that there are cancers that have a similar pattern. Again, there's another group of cancers here that have a different type of pattern, but similar to one another in that group. So for these ones, we don't know what the cause is. All we know is they have a similar process, but this is actually unique information, though. It's really novel. And then we have this group of cancers here that have a particular pattern or profile related to the colibactin damage from PKS coli. So this tells us that within a bunch of cancers, we can identify which ones have been caused by colibactin, which ones have a specific cause that we don't quite know yet, and then which ones have a likely different cause compared to these ones here. So what we did then is the group that we that are identified by the colibactin signature, we looked at in 1,600 colorectal cancers. These are the numbers that we saw it in. So the things that we saw were the ones that had the colibactin signature tended to be diagnosed at a younger age. So the colibactin signature was less likely in people that were older. They were also, the cancers with the colibactin signature were more likely to occur on the left side. And they were also more likely to have one specific type of mutation, which is this mutation here. But the key here is it's in a particular gene, which is really important to the development of colorectal cancer. So colibactin is causing a specific type of mutation in a very key gene that causes colorectal cancer. So what we think is happening, or what we know is happening now from this evidence, we think that there are early life exposures that enable colonization of PKS coli in the bowel. I think they might be happening in childhood or adolescence. Allowing PKS to enter the cell. So this is one microscopic crypt within the normal colon. It gets in there, causes DNA damage at the bottom of that crypt in those cells, and that enables growth to an adenoma or a polyp. And that DNA damage is measurable through this SPS88 signature, and also by hitting a particular gene, and those two combinations resulting in the development of early onset colorectal cancer. So that's what we think, the mechanism is. So what does that mean then? Well, being able to measure and identify what's caused a person with early onset colorectal cancer, what's caused their cancer, is being communicated to us with our people with lived experience. So that's really important to them to understand that they know what's caused their cancer. So being able to detect whether it's come from Colibactin is really important. What we don't know is, does this have a better or worse prognosis if you have this particular type of cancer? And we don't know if it has certain treatment implications yet. So they're things that we need to find out. We also hypothesized that if the bacteria is in the colon, there might be more than just one region of the colon that has had DNA damage from Colibactin. So that may put you at risk of getting a second cancer, but we have no evidence for that yet. And the other factor that we're looking at the moment is, well, if the environment or diet is helping PKS colonize the colon, perhaps that shared diet with your family members may also put them at risk of getting a similar type of cancer. So we've got funding from the Cancer Council of Victoria to investigate these two areas, which will be really important to provide an evidence base as to what we should do when someone is diagnosed with a Colibactin-related early onset colorectal cancer. So for the unaffected population, then it means we should probably start to think about identifying who has PKS E. coli and how we should do this, whether it's through the stool, whether we can use the fecal immunochemical test from our National Bowel Cancer Screening Program to be able to do that, and more importantly, when we should be doing it and how frequently. So these are all questions that need to be answered to enable us to develop a population screening program and prevention program. But knowing more about these areas will also enable us to develop intervention and chemoprevention approaches that may inevitably be able to inhibit Colibactin's DNA damaging activity, or we may be able to eradicate the bacteria, for example. So there's lots of work to do, but it really offers some really exciting areas for prevention and early detection. I want to come back to this figure here of the mutational signature patterns for all these early onset colorectal cancers. And just to focus on this one group here, you can see they have a very prominent signature, which is indicated in the red here, which we call SPS89. And for this particular group, we don't know what causes SPS89. We know SPS88 is related to Colibactin, but we don't know what that is for SPS89. But we're hypothesizing that it might be a different genotoxin from a different bacteria. So to come back to where we were in the beginning with our ring of what causes early onset colorectal cancer. So from this unknown group, we've been investigating potential causes. So we can see that there's this group here from our signature profiling that have this SPS89 signature of unknown cause, hypothesized to be a different bacteria. We have this group of about 5 to 10% of early onset colorectal cancers that are caused by Colibactin DNA damage. And then there's two other subtypes here that we've identified through our profiling. So we're starting to build up a much better picture of the different causes of early onset colorectal cancer by measuring the tumor tissue itself. Interestingly, this green group here, which we called HRD, is caused by inherited mutations in the BRCA1 gene. And many of you may know that gene because it's a breast cancer and ovarian cancer gene, but it's been very rarely associated with early onset colorectal cancer. So we've shown that in a small proportion of cases, people's early onset colorectal cancers have been caused by a mutation in the BRCA1 gene. So that, again, more information about the causes so we can develop prevention and early detection strategies. Then to kind of try and get at the question of, well, we're looking at the causes of early onset colorectal cancer, but what are the causes of the increasing rates of early onset colorectal cancer? So we've mapped our causes here from our signature profiling across the year of birth of the person that developed early onset colorectal cancer. And we're starting to see a pattern that more of the more recent cases have Colibactin and SBS89 causes for their colorectal cancer. So that's given us some preliminary data to work with to not only identify causes, but what's causing the increasing incidence. And that's recently led to us securing some funding from our National Health and Medical Research Council and Cancer Australia to look at what the causes of the incident increasing rates are in Australia. So it's quite a complex figure, but essentially we're taking colorectal cancers that were diagnosed in the late 90s, early 2000s, and comparing them to newly diagnosed early onset colorectal cancers. And we're looking at signature profiles, the microbiome composition, and microplastics to see if there are certain microplastics that are present or more common in the newly diagnosed cases compared to the historical cases, which we're calling temporal signatures, and then looking to see if there are unique risk factors related to those temporal signatures. So that's 3 years of work we're really excited to get into. And lastly, I just want to touch on this tumor evolution and microenvironment work that we're doing, and really just proposing, because it's a new area for us, just proposing the question that we don't know the answer to yet is, how quickly does early onset colorectal cancer develop? And we know for later onset colorectal cancer that it takes about 10 to 15 years to develop from a normal to a precancer polyp to a cancer. So there's two prevailing hypotheses at the moment. First one is that early onset colorectal cancer takes the same time as later onset. It just starts earlier. So exposures that are happening earlier in life are causing the same development of colorectal cancer, and it's just presenting at an earlier age. The second hypothesis is that early onset colorectal cancer is developing really rapidly, less than 5 years' time. So understanding which or both and what proportion of early onset colorectal cancers might develop by either one of these pathways is really important to understand for us to be able to develop prevention and early detection programs. So that's something that we're working away on at the moment. Nationally, we've built a collaborative research network looking at the biology and etiology of early onset colorectal cancer through a Centre of Research Excellence that's being funded by Bowel Cancer Australia, and it's called Artemis, which is the Greek goddess of hunting. So we're saying our Centre of Research Excellence, we're really hunting the causes of early onset bowel cancer. We've had fantastic collaboration and engagement with Bowel Cancer Australia, who do fantastic work in the national community. They've developed a national action plan and roadmap for early onset colorectal cancer, developing clinical practice guidelines specific for early onset, and lots of other activities here, including the first national research symposium for it. Internationally, there's a global early onset colorectal cancer think tank that started in June 2025. You can see 18 different countries are involved. And again, that's really become focused on what's the causes of early onset colorectal cancer. And they developed 4 specific working groups to tackle this issue and to bring international coordination of research efforts to tackling this problem that's now become a global problem. So in summary, early onset colorectal cancer is now really a major public health problem in Australia and across many other countries in the world. And in Australia, it's the leading cause of cancer-related deaths in people under 50. And the projection is the incidence is likely to continue to increase, and deaths will continue to increase because we have no mechanism at the moment to stop this rise. And that's largely because we don't understand the causes, which is something that we're working on in particular, including genetic causes. And I showed you that particular example of this PKS coli bacterium, Colibactin, and how through looking at the tumor itself, it's given us clues as to how that PKS coli and Colibactin has caused that particular cancer. And so there's lots of other questions that we need to develop evidence for to help manage people and their relatives for when they have PKS coli related colorectal cancer. And there's other subtypes that we think have related to other bacteria which we're investigating, which I showed you related to SBS89. There's some fantastic national activities now which engage with funding So we're at the beginning of hopefully some really prosperous years in terms of generating some new knowledge around the causes. And internationally, the community is coming together to work together to find answers, which is a fantastic thing, which will only speed up the process. So I want to thank, in particular, the people in my group and the people with lived experience that work with us to help develop our research program. And also a really special thank you to Julian Wiggins, who's the CEO of Bowel Cancer Australia, who does a fantastic job advocating for people with bowel cancer, but also helping to drive the research agenda. And thank you to our funders, the NHMRC, Cancer Australia, Cancer Council Victoria, and the Cancer Council New South Wales. So I will leave it there, and just to say thank you very much again for the opportunity. And if you have any questions, my email's there, so please feel free to email me with any questions or comments you might have. Thank you very much, and hope you enjoy the conference.